ठीक है, we are live. We can leave it at that. Hi, Rutuja.
Hi, Rituja.
Hi.
Rituja is also trying to keep purple.
Yeah, going with the theme.
Yeah. Let's get people in. Arushi, Rutuja, just 1 minute before you get people in. Okay, we are already live on YouTube. Hmm. Shall we let people in?
Yeah, yeah.
Hmm.
Also, I'm not a co-host yet, huh?
Okay, ठीक है. Then let me make you co-host. Let's just keep letting people in as they come in. Hi, Tripura. Good afternoon.
Good
Hi,
afternoon.
thanks for joining. Thanks for joining. And
Thanks
our
for
first speaker is also here, Venkatesh. Hello, Venkatesh. Good afternoon.
Hello, ma'am. Hello, good
We'll
afternoon.
we'll just start as soon as our other expert mentors are here, and we'll
Okay,
get
ma'am.
started very soon. Yeah.
Thank-
Venkatesh, are you ready with your slides and things
Yes,
like that?
ma'am.
Great, great. Yeah, and really good to see everyone. And we have some returning attendees who have been regularly following up through the sessions. Tripura, we're just waiting on Vidya and Manjari, but we'll get started in a minute. Huh?
No
Tripura and
worries.
Venkatesh, yeah, thank you.
No
Thanks
worries,
for your
Rituja.
patience.
Yeah, no
Yeah.
worries.
Rituja, you just want to send a reminder to the other speakers. I have already sent them, but I don't see them in the waiting room. So you're CC'd on that email. Can you just send them a reminder to please join?
Okay, okay. For
Yeah,
expert
I've
mentors, na?
no, for the other speakers, PhD
Okay,
speakers.
okay.
Yeah,
Great.
thank you. Thank you. They're probably having a comfortable lunch before their presentation. So Tripura, where are you based? You're at Hyderabad.
Yes, Karishma, I'm at CCMB.
Great, great. How's it going there?
All good.
Yeah, Hyderabad is a nice city to live in.
Yes, yes.
Yeah,
You're at
yeah.
Pune, right?
Yes, that's right. That's right.
You have two other colleagues of ours in the panel, Aishwarya and Jyotsna.
Yes, we had Aishwarya yesterday, I believe. Yes,
Yesterday, I think.
yes,
Yeah.
yeah. That's right.
So how are the
Okay.
sessions going?
So far, so good. A lot of feedback from the expert mentors. The talks have been really good. They've been running on time, and a lot audience engagement as well. So far, so
Yes.
good. Yeah,
Yes.
yeah,
I think
yeah.
it's a good initiative, actually.
Thank you. Thank you. Yeah. Um, all right. I think I'll get started, for want of respecting everyone's time. I think people will filter in and we can just catch them up because it's 2:05.
Yeah, sure.
Yeah. So, welcome everybody. Welcome Venkatesh. Welcome Tripura. Really good to have you both. Yeah, Vidya's joining. It's almost as though she could hear me before she was in the meeting. But yeah, Vidya's joining. That's great. Yep. All right. Welcome everyone to PhD Cafe. We have an exciting lineup today afternoon. It's animal biology. And to set things off, it's going to be Venkatesh, followed by Sushmita, Anvesha, Dimple, and Anirban. So welcome to all of you.
Thank you.
We look forward to having you. Um, so the general guidelines is make sure you can hear and see us. The slides will be shown. Feel free to use the chat window for all suggestions, comments, but each talk will be 20 minutes and the expert mentors can directly ask their questions to the speakers. The audience may use the chat window. And so 20 minutes of talk time and 10 minutes of expert mentor feedback and audience feedback can keep coming in through the chat window. So of course, we want to keep the feedback constructive. And as we've been doing through the session, for those audience members who keep the session interactive, we have a giveaway for one member every session. And we had 5 giveaways so far. It's been going well. So thanks all. And these are our mentors for today. Manjari, I don't see yet in the chat window, but I'm sure she's joining. So thanks, Vidya. Thanks, Tripura. Thanks, Manjari, for agreeing to do this. And your role will basically be to listen to the talk and then give suggestions, feedback, inputs, ask questions. So like a pre-defense experience. An audience Q&A would also happen if we have some time. So I think we are all set to go. We will keep time, Venkatesh,
Yes, ma'am. Yes, ma'am.
20 minutes for your talk. So why don't you start your screen share and slides, show us your slides and then we'll get started and my colleague will keep time and also let other people into the meeting room. Yeah, we're also live on YouTube, just letting you know. Great, let's get going.
Wait. Is it visible, ma'am?
Yes, it is. Yes, it is.
Okay, thank you.
20 minutes.
So
Great.
okay, thank you. So good afternoon, everyone, expert mentors, respected expert mentors, and Charisma ma'am, India Bioscience, for giving me and selecting me this for PhD Cafe. So for today's presentation, I'll be presenting my work, some of my work, and that is a tumor-derived osteopontin mediates macrophage infiltration and polarization as a potential target in intratumoral immunity in melanoma. So I am working with Professor Gopal Sikundu, who is Director R&D at KIT University. So we'll get started. So based on what is cancer, so cancer is a disease where non-communicable disease where there is a need of mutation of oncogenes, and for its cell to proliferate abnormally. So this is how Professor Robert Weinberg from Whitehead Institute, what he suggests, he said, at least that need to be 5 different mutant genes to be cell to be proliferate inappropriately. If a single gene is like a mutation in single gene cannot create cancer, if that is so, then we'd be covering throughout all over the body with cancers by the age of 6. So this is the redundant mechanism what happens within the cell that controls each other's the oncogene mutation by the tumor suppressors. So any short circuit happens in this circuit that allow the cells as a whole to proliferate inappropriately. So this is how the cancer initiation happens. So my work is on melanoma. So I work in studying cancer, taking melanoma as a model. So this is the hallmarks of cancer. As all we know that this is generally published by Hanna and Weinberg in various reputed journals, specifically Cell, with time to time upgradations. So these are the hallmarks of cancers. And epidemiology of melanoma coming to this. So in North America and Europe, it accounts 46 and 32, 46.4 and 32.4 respectively. And this data is for Globocon 2020. And this India has in Asia also we can see that Asia has in India 16.5%, but it is very less as compared to other cancers, but still studies need to be done in future so that there should not be any outbreak. So this is melanoma where actually the melanocytes, pigment producing melanocytes, tumor originates from there. And generally it is a mutation, unpaired DNA damage. And the property is also it can multiply rapidly. So what is the reason for this? How the melanoma causes very specifically UV exposure and sometimes genetically predisposition of the disease. So earlier, as most of the cancer in earlier stages, it is curable, but in later stages, it can be like a metastasis to other organs like lungs, liver. So this ABCDE is many of you knowing, but it is like a nursery ABCDE we were thinking, but this is actually what the diagnosis one physician will see whether it's the patient has a melanoma or not. So A stands for asymmetry, B for uneven borders, C for the change of more than one color, D for diameter, E for evolution. So this is how the one can diagnose that is because we have red moles also. So people should not be confused with the red mole and melanoma. So what we are searching is for a novel immunosuppressive secretory factors. So our studies based like we are targeting some of the secretory factors as they are acting as immunosuppressive factors. So this is how the immune checkpoint inhibitors were, apart from CTLA4, PT1, PDL1, there are other checkpoints which has been discovered. But our protein of interest, the gold standard protein, is osteopontin. So this osteopontin, whether fits here or not, so we are just studying that. So now our aim is to target myeloid cells for the immune therapy and prompting these macrophages with specific stimulus for the immunotherapy. So this is the beautiful structure of the osteopontin where the architecture was done is splice variants. Interestingly, you can see that this osteopontin has a wide range of integrin binding domains with the various cells on like T cell, myeloid cells, lymphoid cells, cancer cells. And there's a CD44 binding site, which you can see here. It is a co-receptor. It has also initiate many downstream signaling molecules, thereby promoting the oncogenic functions. Interestingly, this osteopontin has a splice variance. Osteopontin is required for the general physiological process. But what is different is these splice variants are generally upregulated in transformed cells. So this osteopontin A, B, C, 4, 5. And beyond that, recently, osteopontin 5 has a 4 more splice variants, which you can see. And these are different from their exon shuffling and their exon missing. So they are differentiated by this. So this is the osteopontin, where osteopontin, I think how it will be. This osteopontin is born in bone and brought up in tumor. So it is a bone-derived protein, but it is most upregulated in many of the malignancies. So this is how the track record of our Professor Kundu's at NCCS and here KIT University is 30 years of research where he has dissected the molecular fundamental mechanisms in the breast and other cancers. But one step ahead, osteopontin role how we have shown in stromal cells, like you can see this oncogene paper where in 2021 it published by one of our senior, which we can see in the cancer associated fibroblast, also in tumor associated macrophages, also the role of osteopontin has been implicated. So this is the role of osteopontin beyond like osteopontin is not only restricted to tumor cells, but also it has been a diagnostic marker along with diagnostic and prognostic marker. This is a new paper, recent paper was published in ball cancer, but actually the Bill et al. Published in Science. So here CXCL9 and SPP1 ratio determines the TAM polarity. So high SPP1 ratio is a pro-tumor, where the low SPP1 CXCL9 has a high ratio as an anti-tumor, but low ratio as a pro-tumor. So this signaling pathway, which depicts the molecular basic fundamental pathways, which has been discovered in the field of osteopontin. These are the integrins, and these are the oncogenic functions, how the downstream signaling pathways. And this literature also shows how this from our lab, where we have so not only osteopontin act as on the tumor cell, but it's the immune and stromal cell interaction also osteopontin is important. So interestingly, what second thing is the function of OPN in tumor immunity continuum. So we know that tumor immunity continuum, inflamed, immune excluded, immune desert, but inflamed and immune excluded, so far osteopontin role is known, but it is yet to be studied in immune desert. So this is how the potential intratumoral immunotherapy, how osteopontin is responsible, as I have shown that it has a communication where you can see SPP1 positive macrophages, and these CABs has their communication network. Here also this as a FAP and SPP1, they have a like crosstalk, and it is a high SPP1 is less benefit from this immunotherapy. So how this is a study where you can see here the knockdown of SPP1 reducing the down regulating the expression of CD206 at different time intervals. If you take 48 hours, 95% 95.5% to 6.12. So osteopontin has is role in like polarizing macrophages into tumor associated macrophages. So this study in the lung cancer. Sorry, it's not moving. Hello.
Yes,
Yes, you are
Venkatesh.
audible.
Yeah, okay, thank you for the confirmation. So this is osteopontin is a it can be a novel immune checkpoint, where this study is an interesting study where they have a 4 clones of osteopontin. So these when they are treating to like this mice in vivo experiment, so when they did T cell proliferation rescue assay, so this is a high T cell proliferation after blocking the osteopontin. So this is how the literature up to this I have just highlighted. So this is from here my our work on how the osteopontin triggers the melanoma progression to tumor associated macrophages. So this is we have while in plots we shows tumor and normal, the level of expression of osteopontin in tumor is high, but high osteopontin has a poor survival probability. And this CBio portal we have deployed, we have just publicly available databases. So this is osteopontin and CD163, osteopontin with CD206. These are the macrophages, tumor associated macrophages, M2 markers. So there is a positive correlation with osteopontin and these macrophages. So after that, we just check whether this osteopontin has a role in vitro conditions. So we have taken two models, mouse model and human model. So our raw cells were treated with conditioned media B16F10 cell, which is a mutant melanoma cell, and raw is a mutant macrophage. So conditioned media, you can see the morphology change with the dendrites outcomings with this. And here also we can see is a THP1 PMA induced THP1 monocytes induced with PMA. So then A375 is a human melanoma cell line. So there also the morphology is changing. So based on that, we have done the CD206 and CD163 expression. Those are also going up. So here we can say that melanoma derived secretory factors has a role in polarizing or activating macrophages into tumor macrophages. Slide not moving. Slide move.
Are you unable to move the slides,
Yes,
Venkatesh?
ma'am. It's
No,
fine.
it moved.
Yeah.
It moved. Yeah.
So here we have hypothesized whether because of osteopontin, whether it has a role in this activating macrophages. So we have knocked down, silenced osteopontin in B16F10 and A375 through lentivirus mediated knockdown. And these are the confirmed by the knockdown efficiency confirmed by Western and QRT-PCR.
Could you
So
please
here
spoken
what
with-
we have done is we have treated RAW cells with the parental B16 and CM conditioned media of OP and SHB16F10 knockdown. So here we can see with the parental B16 34.88% of CD206 population, but here 14.24%. So it's a reducing CD206 positive macrophages. So similarly, we have treated the recombinant osteopontin with a different concentration of osteopontin human recombinant. So we have checked CD163 also. So there is an increase with the increasing concentration of the recombinant osteopontin. So after that, what we have moved to study these osteopontin role in in vivo models. So we have three groups where we have taken C57 mice where we have injected B16F10 cells, B16F10 plus activator macrophages, again B16F10 plus activator macrophages with OPN neutralizing antibody. So very interesting result. You can see the blocking osteopontin inhibits polarization, macrophage polarization, and reduce tumor growth. So this is a group 1 where only B16F10 is injected, but here with the activator macrophages with the B16F10, and here the neutralization antibody is we have just treated with this neutralized and with the alternative Ds. Then the tumor weight and tumor volume has been calculated. So we can see here there is a very impressive tumor regression in OPN neutralized antibody treatment. So this is how so we can conclude that osteopontin inhibits macrophage polarization. Although our gene expression studies are in the pipeline. So we will be studying with these tumors and various studies we will be doing now. So after that, we are very interested to studying the OPN splice variants in various cancer cell lines. So we have just started this experiments where we have checked the OPN A, B, C, 4 and 5 through, these are the transcript level expression, transcript expression. So A375 is the melanoma cell line, but we have checking as a, this hepatocellular carcinoma, and 231 is a TNBC cell line, and MCF7 in all the the splice variants. So we will be doing that for the further process. So this is the part of another objective where we have just want to show that whether these tumor associated macrophages any role, because that's the ultimate how the tumor associated macrophages enhance the tumor growth of melanoma. So when we treated the M2 M1 and M2 conditioned media to A375 cells, so So we can see that there is where at 12 hour the percentage of migration with the treatment with M2CM is high. Similarly, the colony formation also with the M2 condition media is high. Although the M1 condition media treated are those are is troubling, but these are maybe the due to the pleiotropic secretory factors like IL-6, which has a pro and anti-tumor properties. So we will be doing secretome analysis of both M1's condition media and M2 condition media, and we'll be finding what are the upregulated secretory factors. So this is how we has just moving for the osteopontin mediated TAM, osteopontin and tumor mediated melanoma growth. So this is a summary. So melanoma is all we know is an aggressive malignancy, but our preliminary results that the OPN drives the resident macrophages to TAMs through tumor stroma interaction. So we found that OPN preliminary results, we found that OPN has a defining macrophage functional states within the TME. So our suggests as the OPN triggers tumor cell migration and proliferation. So the underlying mechanism studies are in the pipeline to study the macrophage functional state. And so we want to confirm that whether we wish to confirm whether OPN act as a double-edged sword in tumor microenvironment. So these are the some of the publications. And I'm thankful to India Bioscience and the company of biologists for giving me the opportunity to present my work. And I thankful to KIT University and DST Inspire for providing me fellowship. Thank you.
Sounds good, Venkatesh. Well within time. And why don't we take some questions from
Yes,
our
ma'am,
expert
definitely.
mentors? Sure.
Yes.
Go ahead, Vidya, Tripura. We have some time because the next speakers have not joined for whatever reason, in spite of multiple reminders. So take your time over the questions and answers.
Okay.
Yeah.
Okay, Venkatesh,
Yes, ma'am.
Go ahead, go ahead.
that was oh, sorry, you were going to start with you. Sorry. Okay, so that was initial good work, but I have some questions before I would give some suggestions. So you're looking at the that the secreted media is actually that is what is activating the macrophages. So that is the optineurin present in that is what is the from secreted media it's coming, and that's what is getting activated, right?
Yes, ma'am.
Did
Yes,
you
ma'am.
measure the concentrations of optineurin present in that at the expression level, and at what concentration is it needed?
Yes, ma'am. In the conditioned media where it is like we are treating with the without like FBS, so it is an incomplete media. So we'll be checking that also. So the concentration is very important. So we'll be checking the we'll be checking the actual concentration at what concentration it is converting the polarization. That's a good. Yes, ma'am.
Yeah, so you're keen and you're very sure that there is optineurin, that's why you're seeing that polarization, right?
Yes, ma'am. Yes, yes, ma'am.
So other than this, maybe it would be a good item. You already mentioned you'll be doing the secretome analysis. So
Yes,
if the secretome, you want to go ahead with the proteome, or you want to do the RNA-seq as well?
Yes, ma'am. RNA-seq we are planning. So we have done also at NIBMG RNA-seq. So we have the analysis is going on. So after we get that, we'll be seeing what. So secretomics is not yet started, ma'am.
Yeah, okay. Could you just go back to your mouse model? I couldn't follow it much there.
Yeah, just I just stopped here. This visible, ma'am?
Yes, it's visible. So could you just tell me what you're injecting? Where are you injecting this in these mice? And these are human cells? No?
No, these are mice cells. B16F10 is a murine melanoma cell line.
Okay, it's in the melanoma cell line.
Yes,
No.
ma'am.
But with this, I mean, I don't know how good it would be. Normally, we do these experiments in skid mice, so the tumors are much more, clear and evident. Since you
Yes,
need
ma'am.
maybe because it's not quite clear in this on how. So it is the is it a subcutaneous kind of a tumor that's developing there?
Yes, ma'am. Yes, ma'am.
And you're
Subcutaneous.
excising that part out and to show it.
Yes, ma'am.
And
exactly.
this color, I was not able to follow why the tumor color, is it just along with the skin and
No, so actually there's a melanoma. So as is a
right.
melanoma. Yes, ma'am.
So you remove the hair over there, shave it up, and then you take it out, looking at.
Yes, ma'am.
Okay, fine. I couldn't quite follow this one. And that was my main suggestion, that it's important to look at these exosome or the microRNA data. Maybe you could also focus if the optineurin is secreted directly, or is it moving more into the exosome fraction, which
Yes,
fraction
ma'am.
of the secretome. That would help you understand how is it, maybe is it carrying alone, or is it carrying some other factors along with that. Maybe that would be a point you could look at as well.
Yes, ma'am. Definitely, I will go that.
Yeah. Yeah, I'm done.
Thank you, ma'am.
Okay, this is not related to my area of work at all, so I'm just going to ask you something very general
Okay, ma'am.
and give you some general comments. I was wondering why you are interested in osteopontin at all, given that multiple genes and proteins are implicated in cancers, right?
Yes,
From past literature. You've also shown some of that. So I was wondering, why were you specifically interested in this and not in something else? I mean, it
yes,
would be good to give some of this background when you are presenting.
yes, ma'am. So in introductory slides, which I've shown that osteopontin is a like bone derived protein. So
Correct.
it's a physiological process. It has a tremendous role because it needs to be
Right.
optimum concentration to maintain the physiological. What is
But that is true of so many other proteins and
yes,
genes.
yes.
Yeah.
The interesting is that the splice variants which osteopontin has. So
Hmm.
that splice variants has a interesting role in, because we can see the osteopontin C is highly expressed in breast cancer, but it doesn't express in lung cancer. Is the osteopontin B highly expressed in lung cancer? So these the dynamic nature of the splice variants can, like, we can target this splice variant with a specific will like small molecule inhibitors or various combination drugs, so we can just target the cancers. That's the reason. And yes, ma'am, I can say the osteopontin. Why osteopontin? Because as I can say that the lab I joined is Professor
Okay.
Kundu has
Works on. Okay. Fair enough. Okay. But now, given that you said that blocking osteopontin inhibits macrophage polarization and therefore it might be uh, useful as a target,
yes, ma'am.
but given that osteopontin is also required for normal functioning, how exactly do you plan to
Yes.
go forward with this?
So that is the beyond the threshold concentration, whatever whichever the protein, like these oncogenic proteins are expressed in transformed cells. So that's the how we will be moving forward for designing any drugs or anything targeting osteopontin. So we need to find it out how we will be designing those. Is it the full length osteopontin will be targeting, or the splice variants will be targeting? So that's the way our approach will be.
And at a particular threshold that is not going to interfere
Yes, ma'am, with the
with
future.
normal functioning. As a general comment with your presentation, I think you can leave out a lot of your initial figures that you're not explaining, figures from past work, because there are a lot of these figures with a lot of stuff which we can't really read in that shorter time.
Yes, ma'am.
And you don't have the time to get into any of those either. So it might be a good idea to get rid of a lot of them and then also maybe do some spelling and grammar checks in your text.
Okay, ma'am. That's in a hurry. So because of
Yeah,
time.
sure, sure.
Yeah.
No, I mean for your final.
Yes. Okay. Thank you, ma'am.
Adding
All
to that, point is that, exactly that even I was having this doubt, why osteopontin, I could just understand because it's in your lab, but you have to give little bit
Yeah.
of more introduction on osteopontin and its general function. We all understand it's in the bone development, but that would help more for a wider audience to really catch the point.
Yes.
That was the first thing which was sitting in my head, but then as I saw that is from the lab previous work, you were showing so many slides. So ma'am is absolutely right. So it's better that, you add more introduction on osteopontin first, justify why you took up this problem, that is fine, but why osteopontin itself?
Yes.
And melanoma, also, like you've already mentioned, though it is very less, you may find it a little difficult to defend in the long run, saying that though it is not such a major cancer
In
in
India.
so you
Yes.
will need to find out a very strong reason. No worries, you should continue any amount of thing. We have a problem, we should be working on that. But for you to build on more and add some justification to why you chose this particular problem, it will
Yes.
just help in clarity. That's all.
Yes. Thank you. Definitely I'll work.
Great points. Any other points, Tripura, Vidya?
And
Excellent. Anything, just that maybe we can, the new, next speaker has joined, Sushmita. Sushmita, are you there? Sushmita Nandy? Yeah, I can see her in the meeting. Hi, Sushmita. As you get ready, I know you've just joined. It's like running into a meeting room physically and then having to immediately open your laptop and giving a talk. So just settle in. We will give you a few minutes. So, Venkatesh, which year of your PhD are you in?
I'm in second year, ma'am, completing the second year. Yes.
Okay,
Yes.
that is remarkable to have a
Ah,
oh,
full-fledged
okay.
talk.
I thought only
Yeah.
senior PhD students are allowed to
So
give this.
prioritized senior, Vidya.
Oh,
But
I
if
see.
we didn't get enough seniors, then we had really good talks from people earlier. We did offer them an opportunity. So maybe
Oh, I
Venkatesh is
see.
one of them.
Yeah, so
Okay,
that
okay.
explains
Okay,
the
then that makes sense.
yes.
Yeah. Well done, Venkatesh. So
Thank you,
any
ma'am.
questions from the audience for Venkatesh? Any other questions?
One more
We do
point I
have a,
want to add
sure,
that,
go ahead.
yeah,
Go
because
ahead.
osteopontin, why osteopontin? Because we are just trying to that LILRB4, which is Professor James P. Allison at MD Anderson, he is working on. So through this in silico studies, what we can see that this LILRB4 has a positive correlation with osteopontin. So those this is a novel immune checkpoint what Professor Alishan is studying. That's a paper published in JCI in 2019. So this is so we are working on that also. So our project is decided. So sir will take it forward.
Further, that makes sense. Great. And Sushmita, are you getting ready?
Yes, ma'am. I'm
Great.
ready.
Thank you, Venkatesh. Wonderful talk. Thank you.
Thank you,
And
ma'am.
thank you, Vidya and Tripura. Great. Sushmita, we can see you. I think we can hear you. And now the question is, we need to make the slides work.
All
So,
the best, Venkatesh.
yes, all the best, Venkatesh. And all the best to you, Sushmita. Please show your slides, Sushmita. Can you start screen sharing your slides?
Okay. I think actually I am sharing the slides with you yesterday.
Okay, you
Share?
want us to show the slides?
Yes. If
Sure.
you
We can do that too. If you've shared it, we
okay,
can
please
do
share.
that.
Okay. Okay.
Sure. I'm sharing it. Okay, Sushmita? All right. I've opened your slides. They look good. I'm going to share them. And Sushmita, then you'll have to tell me next karke, right?
Okay.
Can you see your,
Definitely.
Sushmita, can you see your own slides?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Great. So just tell me when you want to move next.
Okay, ma'am.
Go ahead, start. Yeah. You have 20
Okay.
minutes,
Okay.
Sushmita. All the best.
Okay. Okay. Good afternoon, everyone. Today I'm here to present my work on the title of a comparative study of islands protein alpha crystalline in different ages of rohu fish and recombinant human alpha b crystalline. I am Sushmita Nandy, research scholar in the Department of Biotechnology of St. Xavier's College. Now I am working on alpha crystalline. Ma'am, please next. I am working on alpha crystalline. Here is it is the major class of predominant islands protein among all vertebrates, and this is the structure of monomeric form of alpha B crystalline. Here only ACD domain is shown, that is alpha crystalline domain, and the major classes are alpha, beta, and gamma crystalline. Alpha crystalline possesses of two subunits, alpha A and alpha B, and it has three domains: N-terminal region, alpha crystalline domain, and C-terminal tail. The concentration of alpha crystalline high in lens cortex but low.
Sushmita, have you dropped off accidentally? I can't see Sushmita. Can you see her? Anybody?
I see her in the meeting, but we
No,
can't see her.
But video suddenly went off, no?
Yeah.
Yeah. Hmm. Maybe we'll just give it a couple of minutes for her to join back. I don't see her now in the meeting room either, Arushi.
Sorry, ma'am, there was some
No
internet
problem,
connection
Sushmita. No
issue.
problem. No problem. Just calm down. No problem at all. Your slides are there. We are there. Go ahead. Yeah.
Okay, yeah. Now, continuing this, alpha crystalline composed of two subunits, alpha A and alpha B crystalline, and three domains: N-terminal region, alpha crystalline domain, and C-terminal tail. The concentration of alpha crystalline is high in lens cortex but low in lens epithelium. It possesses small Ichthyoc protein having molecular chaperone activity. It can prevent cataract, and it provides thermocellular resistance. Next slide, please, ma'am. Now coming to my next slides, that is the objective parts to show the conformational changes of alpha crystalline in native and denatured state. And second one is to observe the biophysical structure and functional changes of different aged rohu alpha crystalline, to differentiate between human and fish by establishing the phylogenetic relationship with recombinant human protein, to determine the relationship between such age-related changes and chaperone activity of alpha crystalline, to observe the exact oligomeric size and radius of the alpha crystalline in fish and different ages, and to assess the anti-aggregation and repolling properties of alpha crystalline. And one of the objectives are shown in the next slides. Next slide, ma'am. There are some there are some literature references I have used for my work, and these are the some screenshots of that papers. Next, ma'am. For my study, I have chosen the rohu fish of different ages because it is freshwater fish, easily accessible, cost effective, and common edible carp. And the age specifications are 1 year, 3 years, and another year I have taken. But the work is going on. It is in progress. So that's why 1 year and 3 year I have written in my slides. Next one is recombinant human alpha A and alpha B crystalline are used for comparative study. I have taken for the fishes from the Godibari aqua shop located in West Bengal, and they are feeded by Nutrafeed F430J advanced formulated feed, and the breeding is natural. Most important, the age determination by annually count from the fish scales. In the picture, I have shown the scales annually. Only one annually means it is of 1 year aged fishes, and in case of 3 years, it is three annually. And these are the pictures, and the in my left corner, the feed, the food which is feeded to the fishes. Next slide, come. Okay, methodology. These are the methodology part I have used to purify my fish alpha crystalline protein and recombinant human alpha B crystalline protein. The methods are taken from these references: Wages et al. 2013 and Grayling et al. 2009. for different samples. First of all, I am discussing about fish alpha crystalline. The eye lenses were crushed and homogenized in freeze paper, and these are the pictures I have given. And then alpha, beta, and gamma crystalline are separated by size exclusion chromatography. And after purifying the alpha crystalline protein, it is dialyzed, and the dialysis protein is aliquoted in the small aliquots, and the purity of the protein is checked by SDS page, whereas the a small band, that is 20 kilodalton, which is the monomeric size of the alpha crystalline. And in the chromatogram, you can see a peak between 25 to 37. There is a huge peak which signifies the alpha crystalline protein. Next, I am going to the recombinant human alpha B crystalline protein, that is the component cell E. coli BL21 and alpha B PET20B was taken, and through agarose gel electrophoresis we get confirmed, and after that transformation and overexpression with IPTG was done, and then several treatments like PMSF, protease cocktail inhibitor, DNase lysozyme, etc. was added, and the soup collected from these different treatments, and it is loaded into the column to get only the alpha crystalline protein, and the peak that signifies the alpha, purified form of alpha. Next slide, ma'am. Ma'am, one request, can I get some more time? Actually, there was some internet issue, so can I get, ma'am? I
Sorry,
didn't.
I said let's move on. Let's see how much more time, because there
Okay.
are other speakers lined up now.
Yes, yes,
You
yes.
let's see how much more time. You go ahead.
Okay. Yes, ma'am. Thank you so much, ma'am. Now coming to the analysis part, there is different studies I have done for my study. One is BCNS binding study, which is structural analysis. Here I have taken different samples. One is native alpha crystalline protein, which is the native form of the protein, and one is preheated. That is, it is heated for 15 minutes and at 90 degrees centigrade, and then cool it for 1 hour. That is the purified preheated form of the alpha crystalline protein. And in case of 1 year, 3 years, it is shown from the graph that preheated form of protein has more binding intensity than the native form of alpha crystalline, and BCNS binds in the hydrophobic sites of alpha crystalline. So this graph shown, this is time independent kinetics, whereas a significant difference is shown in case of native and preheated form of recombinant human alpha B crystalline. But in time dependent kinetics, between 5 minutes, 30 minutes, and 24 hours, during time, with time and upon heating, the binding intensity in the hydrophobic sites of protein get increased. In case of it is most in case of human recombinant human alpha B crystalline, and it is least in case of 1 year and 3 year species. Next slide. Next slide. Now coming to second structural assay, which is urea unfolding refolding assay. In this study, first of all, the native form of protein is unfolded by treating it with urea, and then it is refolded by removing the urea by dialysis process, and it is done for all the samples, 1 year, 3 years, and recombinant human alpha B crystalline also. And urea unfolding refolding study determines the stability of the protein. In the experiment, we can show, I can show that the rate of unfolding is much higher than the rate of refolding, and from this the stability of the protein can show, and these are the delta G values of alpha B crystal recombinant human alpha B crystalline, human 1 year and 3 year alpha crystalline, and it is shown in the calorie per mole unit. Next one, please, slide, please, ma'am. Next, another functional assay is lysozyme aggregation study, which is the aggregation, which signifies the aggregation of the lysozyme in presence of DTT, and alpha crystalline can prevent the aggregation, as we all know. In case of in this graph, the first line signifies the lysozyme treated only with DTT, and the first line is we obtained, and the second line when lysozyme is treated with DTT along with alpha crystalline to show if the aggregation of lysozyme can be prevented by alpha crystalline or not. But and the results we obtained that in case of 1 year rohu fish, 1:8 ratio of protein lysozyme and protein, the percentage of prevention of aggregation is 81%, and in case of 3 years fish it is 74%. But recombinant human alpha b crystalline, it is 82% in 1:1 ratio. It From this study we can conclude that alpha b crystalline has more chaperone activity and it is more stable protein. Next slide, ma'am. This is my last essay slide, that is dynamic light scattering DLS study, which is also a structural analysis. In this study we can calculate the hydrodynamic diameter by giving the sample by scattering the every particles of the samples in the DLS machine, and the diameter I obtained, that is in case of 1 year it is 4.442 nanometer, and in case of 3 years it is 5.97 nanometer, and in case of human, which is 11.91 nanometer. As we all know, recombinant human alpha b crystalline has the radius is along um, uh, 5 to 7 nanometer, which is half. And I can calculate, and in this slide the diameter is given, and the dh means diameter, hydrodynamic diameter. Okay, next slide, ma'am. Now this is my conclusion slide. From this, I have already concluded that human alpha B crystalline have that a significant difference is observed in recombinant human alpha B crystalline, whereas a low difference is observed in fish crystalline. Human alpha B crystalline is more stable than fish alpha crystalline, and 3 years raw fish is less stable than 1 year. And from lysozyme aggregation assay, the chaperone activity of 3 years is more than the 1 year, and the oligomeric size of human alpha b crystalline is 11.91 dm, means diameter in nanometer, and in 1 year it is 4.442 nanometer, and in 3 years it is 5.97 diameter in nanometer. Overall, we can conclude that fish protein is more prone to aggregate than recombinant human alpha b crystalline, and it is less stable than recombinant protein, and human alpha b crystalline has better chaperone activity than fish alpha crystalline. Next slide, ma'am. There are some future plans I have planning to do in later, and there another selected age of 6 years rohu fish for my study, and to analyze the secondary and tertiary structures. CD spectroscopy will be performed to check the chaperone activity. Heat aggregation temperature control assay will be performed later, and to measure the exact oligomeric structure, refolded protein of the refolded protein DLS will be done, and last but not the least, the N-terminal sequence of the pure alpha crystalline protein, its sequence homology with human can be studied in later. Last, this is my last slide, that is references. These are the references I have taken from my study, and now I would like to convey my gratitude towards my respected principal, Reverend Father Dr. Dominic Xavier SJ, and my PhD principal supervisor Dr. Shudipa Shah, and co-supervisor Dr. Shravani Karmakar for guidance in my work, and my special thanks to India Bioscience for giving me such an opportunity to present my work. Thank you so much to all for listening to my work.
Thank you, Sushmita. Very energetic talk. And great that you managed to connect and the connection stayed. So Tripura and Vidya, please give your inputs to Sushmita.
Sorry for the misconnection. Actually, it was due to in Kolkata there is rainy season, so
I see.
it is raining outside, so that's why it was a problem. I'm so sorry for the misconnection.
No problem.
Still,
No
there is
problem at all.
3 minutes there, so you
Yeah.
finished well ahead of your time, sir.
Great, great. Go ahead, Vidya and Tripura. Great.
Any
Okay,
questions?
I'll ask you some general thing again. Why were you interested in this, in looking at alpha crystalline in
Alpha
Rohu?
protein.
Huh. Why were you interested in doing this?
Sushmita, can you hear
Did you
Vidya's
hear us?
question?
No, no. Actually, I didn't
Oh,
hear
okay,
So,
okay.
her.
I was wondering why were you interested in looking at this alpha crystalline in Rohu in the first place?
Actually, it is a predominant protein found in all vertebrates, and it is
Correct,
present in eye. Actually, it is
correct,
mainly
correct.
found in eye,
Yes.
and we all are interested in the human body science. That is physiology of the human body. So that's why I choose the human alpha
So,
crystalline topic. But in lab it is impossible to work with the human samples, so I choose the rohu fish as a model sample
but do you
for
think
my
it's a good
work. And it,
do you think it's a good model to
actually
study?
fish is the fish resides very lowest. Fish is the lowest primitive, that is lowest vertebrates in the
No, 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 no,
evolutionary
no,
structure.
no, 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 no. Let's not go there. It's not the most primitive because whatever species we have right now are derived. So the
Actually,
ancestors of
several
fish
work
in
was done on zebrafish, so that's why. But zebrafish in India, especially in West Bengal, it is impossible to work with zebrafish. That's why I choose the rohu fish. Why? As it is easily accessible in West Bengal, so that's why
no,
I
actually
choose the.
the reason I was asking the question was because you said that this has already been done in zebrafish. So I was
Yes.
wondering why you chose Rohu. What is it that Rohu is going to give you that the zebrafish study did not?
Rohu is aquatic fish, and it is actually it is marine water fish, while it is freshwater fish. Rohu is freshwater fish. A few studies were done in freshwater fish like katla, but no study was done in rohu, so that's why
Correct.
I.
But we want, if you are interested in the human alpha crystalline,
Hmm,
and this has already been done in zebrafish,
hmm.
what is Rohu adding to that?
Uh, Actually, rohu fish, some work was done in rohu, and I am interested to show if they if
See, for instance, potentially, given that this work has already been done in zebrafish and in some
hmm,
other fish also, you said, right?
no, not necessarily, not actually. All the assays was done in zebrafish.
No, no,
Some.
that is fine. So, given that this has been studied in in how many fishes these has this alpha crystalline been studied?
Zebrafish, dogfish, catfish, some types of
So
five or
several.
six fishes, five
So
or six
let's
fishes.
say several fishes it has been studied in, right? And then now you did this for Rohu. It might be interesting to compare these across the different fish instead of comparing it with humans, right? And then you can look at what the difference is in ecology or
That that's why
evolution.
I choose, ma'am, recombinant form of alpha B human,
No,
recombinant
not
form of
human. No, no, no.
to
I'm saying
compare the.
no. What I'm saying is forget the human. See,
Okay.
comparing one fish and one human, is not really meaningful, right? There is going to be some
Yeah.
difference between species,
Yes,
right?
yes. That's
So
fair.
comparing humans versus one fish species is not really going to tell us all that much. But on the other hand, if you already have data from other fish which are published, then it might be worthwhile comparing your data with those other fish
There
and
are
look at what
some
the
differences.
look at
There are
what the
some.
ecology is of these different fishes, and then see if you can say something about why what you find is different from those other fish or similar to those other fish,
Okay.
and then set that up in a phylogenetic framework, because you can't look at phylogeny with two species, one being a human and another a fish, right? But if you have several fish species, you might be able to add something more meaningful than your
Yeah.
human fish comparison,
Okay, ma'am.
right?
Yeah, ma'am.
Also, what are the sample sizes you have for the different ages of fish?
In case of 1 year, it is more stable than in case of 3 years.
No, no, the
But
sample sizes. How many fish did you sample of each of the ages?
for each year, I have chosen five samples, five fishes, to summarize all the proteins. Because in case of one fish, two lenses, which is quite smaller, but in five fishes, it is 10 lenses, and I have collected all that and crushed them, and a minimum
Oh,
30
okay, okay, okay.
40
Okay.
milligram
So
of protein I have collected from each aged fishes,
so
and
is
then I am
it
working.
it's the pooled crystalline that you're looking at. You don't have any
Cold,
measure of
yes, ma'am.
variability?
Yes, ma'am.
Do you have any measure of variability?
Variability, no, ma'am.
It might
I
be
have
useful
not
to
done.
have some variability, no, so that you know how different individuals that are 1 year old are varying, or at least how different sets of these fishes that are 1 year old are varying. Otherwise, it's just one data point versus one more data point.
Okay, okay, definitely, ma'am.
Okay?
Okay, I will remind. I will
Yeah,
remind it. I will do it later.
yeah.
Okay.
Yeah, Sushmita. So most of my questions were asked by Vidya, the initial doubt which I had. Thanks for covering them, Vidya. It was the first doubt which I had. Why did you choose Rohu at all? And I think, as Vidya was suggesting, you should do across fishes. There's no way you compare a fish and a human. And alpha beta crystalline of human is very well characterized. Everything is done. So we know a lot of information. Unless you want to use it as a model when you want you I remember you showing you want to do crystallization, right? At that point, when you want to compare, at some point you can use it, but not in these initial studies because your experiments will not give you any meaningful information when you're comparing and a fish and a human protein, and that too a cloned protein. It's a cloned expressed protein. Okay, there is a difference between native and cloned expressed proteins because this protein would have been actually it would have been added some mutations to enhance its concept. So I will not comment on the clone which you have chosen, but the idea is that you should first go across species. And second thing is when you extract the protein and when you're extracting all the different crystallines, right? Because you're taking a crude extract. You have all the crystallines in that. In order to show what is the size, you need to do a Western blot. Okay.
Yeah, actually, this is in my list, which I will perform later.
You will
Uh,
have to show that evidence that because doing running a crude extract on SDS page will not be enough to show that you have the protein in there.
yes, ma'am.
So your
Yes.
purification will also not be valid. So you will need to do Western blot for all the three crystals that you are showing, right? And if I remember your data that you're showing, this extract, did you separate out all the three different crystals from the fish extract?
Excuse me, ma'am. Please, can
So
you?
it is you took the entire eye lens and then you extracted the crude total protein. From that you showed you did purification by HPLC, column purification,
No, size exclusion chromatography.
size exclusion. Fantastic. So you took different LUs, right?
Fractions.
Based the fractions based upon this size, and you run the fractions and say so when you did this assay, these different assays that you showed, you did with all the three crystallines separately, or I couldn't see that difference. You showed only you took only alpha crystalline. Only one
Only
you.
alpha.
Okay, fine.
Only alpha.
Okay, so as Vidya has rightly mentioned, 2 points there. One is you need to have variability. So supposing if the protein amount quantity is less, and you may need to take so if you take 5 fishes giving you enough protein, that will be a sample one. The sample
Okay.
two, you need to have another set samples, at least minimum three for statistical variation to compare your data.
Okay.
That
Yeah,
is
yeah.
important. And another concerning point for me is you're buying these from an aqua shop,
Hmm.
right? In this aqua
Yeah.
shop, you don't know in what conditions the fishes they're growing there for a year, right? You're
I
not.
uh, I went there and I checked how they treated their samples, fish samples. I talked to them, and they showed me the food samples, the temperature of the how the temperature of the berry, that is the breeding site of the fish, and how it is well maintained. Actually, it is covered up by the bamboo sticks, so that's why no much more UV rays can insert into the water. So there are well treated pool, well treated the berry. So that's why, and to get confirmed that it is 1 year, I have read several papers that how to confirm the age of the fish. That is, they say the annually count, and I did that. If it is one, in case of 1 year fish, it is only one annually. So that's why I get confirmed that they are correct
No,
because
no, I'm not having debating about the age. All I'm saying is I'm talking in terms of your data generation when you're doing. In general, we do controlled experiments when you're doing.
yeah, yeah.
We have them in controlled lab environments where you'll be able to have a data which is more replicable. When you're doing, all the more reason
Mhm.
you have to do multiple way, because not just three, you will need to put five plus five, so multiple things, because you're doing it from a
Yeah,
you're buying them from
very.
getting them.
Mhm.
You have to do multiple things to confirm. And one thing I couldn't follow is in your DLS data, you're showing that the diameter is different. Okay, understandable. 1 year, 3 years, there's a difference, and with the human, obviously, it is different. What is the
Obviously.
interpretation from this? That what is the conclusion? Why was that done? What was the interpretation for that experiment? I
Actually,
couldn't follow.
DLS study was done to determine the oligomeric structure. As we all know, the alpha crystalline is oligomer, and to know the exact oligomeric size of the protein, we did the DLS study, and here is only the native form of the protein. But when it is under stress, that is when the protein is refolded, the diameter should be changed. But I haven't done that work. Actually, this is my PhD work. I am doing small amount of work, and I am showing that to you.
Okay,
So I will
sure.
do
So you will do
actually.
that, right? You're going to add that information and draw a proper conclusion.
Of
But
course, of course.
I agree with Vidya that you should compare between another fish which is already reported, but not compare, because your study at the end of time you'll not be able to draw a conclusion out of it. So all the hard work will go waste, right? Which year
Yes,
of your
yes.
PhD are you in?
Dr.
Which
Shudipar.
year? Which year are
Oh,
you?
yeah. I'm third year now.
Third year. So I think you still have time.
Recently my registration has done, and I'm third year.
All right. And another final suggestion is zebrafish is very, very easy to cultivate in the lab. I'm pretty sure you'll
Yes.
have some labs there which will have. It's much more easier and much simpler model to work in the lab. So explore that option as well, where you'll have your data more, when you're doing this kind of a study, where you'll have the crystallization and all, you'll have more uniformity in the results so that you'll be able to conclude properly. That's the
Okay.
suggestion for
Okay.
you.
Thank you, Sushmita. Thank you, Vidya, Tripura. This was the whole idea that Sushmita, to really get good feedback, mid-course corrections can be made. So thank you for putting yourself out there so early in your PhD. Well done, Sushmita. Well done.
Thank
All right. Okay. We'll now move on to our next speaker, which is who is Anvesha. Anvesha, you're here, and Vidya, her talk. is probably up your alley. Indian chemical cues on the behavior of the Indian ant,
Yes,
Diacama indicum.
yes.
Yeah, yeah,
Thank you, ma'am.
yeah, sure. Animal biology is so vast. It's microscopic, macroscopic,
Correct.
organismal, molecular. So it was a mix. But all the best, Anvesha, all the best.
Thank you, ma'am.
Thank you.
May I give, ma'am? Okay.
Yes, Sushmita, yes. Anvesha, share your slides, please. Yeah,
Sure.
good.
So is my screen visible? Yeah. Okay. Hello, good afternoon, everyone. I am Anvesha. I work with Professor Sumana Annagiri at the Ant Lab at IISER, Kolkata. My PhD thesis is based on the impact of chemical cues on the behavior of an Indian ant, Diacama indicum. Now, ants, along with termites, wasps, and bees, belong to eusocial insects, which means they are truly social as they have reproductive division of labor, cooperative brood care, and overlap of generations. My model system is called Diacama indicum, which belongs to the subfamily Ponerini of ants, and they are primitively eusocial, which means the colony members are morphologically similar to each other, and the reproductive individual is not physically distinguished from the other workers. But under the microscope, she can be distinguished by the presence of a gland called gemma, and we call her the gamergate. The colony size of this ant ranges from 12 to 261 adults living together in a colony, which is actually small in terms of ants, which can range up to thousands, millions of individuals living in one colony. For our experiments, we go out in the natural habitat and collect these colonies and bring them to our lab and house them in artificial nests made out of plaster of Paris. This is an image of the ant colony we house, and these are the various stages of brood which they have within the nest. In order to do behavioral observations, we need to identify each and every ant differently. But since all of them look similar, we use a method of unique marking them on their body using tester's enamel paint. And this is an image of a marked ant, and this is how a marked colony looks like. Now, under the microscope, we identify the reproductive individual called gamergate, which is present only in one colony. So, she has a gemma and the worker does not have a gemma. Now, unlike us humans who perceive the world with our eyes mostly, which is visual, the world of ants is mostly chemical in nature. And since these ants live in an environment which is closely around the human habitat, they frequently encounter various contaminants which are anthropogenic stressors, and some of which might be pesticides. So, in my thesis, I wanted to study the impact of various chemical pesticides on their colony living, as well as different nest relocations and memory. In brief, I studied the residence time of these ant colonies, which means how long an ant colony lives in one nest, and whether presence of contaminants in the environment will impact how long they stay in one nest. Secondly, I also studied the impact of various chemicals such as a pesticide and a herbicide on their colony living, their nest relocations, and on their memory. Apart from this, I also wanted to see how local farmers use the various pesticides in the field and what are their actual dose-dependent strategies, and also their perception about these insects, especially my model system. And also, I have studied the characterization of intrinsic chemical cues, which is apart from the chemicals present outside, the ants have chemicals which are hydrocarbons present on their cuticle. And these chemicals are important for nestmate recognition or pheromones. I characterized these in my species. Now, for today's talk, I'll be focusing on the impact of pesticides. We humans do not like most of the pest species, but
Among the 9 million species of insect living in this world, only a handful of them are pests. The others are non-target, but the application of chemicals such as pesticide impact even the non-target organisms, and hence Diachama indicum, not being a pest species, is a model system to study how this pesticide affects their behavior in terms of colony living, nest relocation, nest choice, and memory. So my choice of pesticide is cypermethrin, and I'm sure most of you have used this Lakshman Rekha to eradicate the cockroaches, and cypermethrin is the primary chemical. This is a neurotoxin which impacts the voltage gated ion channels and thereby modulates the nervous system of the insects and causing mortality or different behavioral effects. I am using this particular brand of chemical because this is mostly used in the field in West Bengal, and 46% of farmers in West Bengal use cypermethrin. In the first objective, in order to test what is the effect of pesticide on colony living, we tried to simulate natural conditions in which we had a plastic box lined with soil and we sprayed the pesticide at the recommended dose, which is generally written on the bottle, and we kept the colonies in this setup for 5 days. And we tested for colony size, mortality, and the status of the reproductive individual. We found no significant difference between the control and treatment. But we identified 5 pesticide-induced behaviors in the treatment which was absent in control. These are appendage shaking, staggering, in which the ant is unable to move in a straight line, trembling, in which the ant will have problems in her body to move, twitching, in which it will show vigorous and erratic movements, and finally paralysis. Paralysis is a death-like state, and it will be similar to death, but ants generally recover from this. So these pesticide-induced behaviors were absent in control colonies, and this was generally due to the neurotoxic effect of the pesticide. In the next step, we wanted to see if different dose concentrations of this pesticide would impact their behaviors, the 5 pesticide-induced behaviors, differently, and also impact their mortality differently. But why did I use so many different concentrations? Because in field, farmers generally do not follow the levels of the pesticide, but use it in a very not dose-dependent way, and they might use more or less. Hence, I chose 4 times, 2 times, half, and 1/4 of the recommended dose along with the control. And in this experiment, I did 3 hours of focal behavioral sampling for each colony across 5 days. So if you look at the graph on the right side, it shows in the y-axis you have mortality, and with different colors I show the doses across the days. And 4 times the recommended dose has caused significant higher mortality and on day 5, which is approximately 12% of colony death. In terms of behaviors, on the lower graph, we can see on the y-axis the average colony individuals who were impacted by these behaviors. And on day 1 and day 2, highest behaviors were seen. But eventually, when the days increased, the ants generally could metabolize the toxin and recover from the behavioral effects. Now, imagine a situation where an ant colony is living in nature and their nest is destroyed. They will have to relocate from their old nest to a new nest site, which is by the mechanism of colony relocation. In our species, these ants do not lay any pheromone trails, but use a method of recruitment called tandem running, in which an individual who knows where is the target nest will come back and call her nestmate and take her physically from an old nest site to the new nest site. The individual who knows where to go is called the leader, and who is following her is called the follower. So in this video, we can see the ant who is going in the front is the leader, and we have the follower. So this is a method of recruitment behavior in diachama indicum, and in order to understand the relocation dynamics, we measured 3 time points, which is the discovery time of the new nest when it was discovered by an ant, the time duration from this discovery to start of one tandem run, and finally how long did the whole colony take to move from the old nest to the new nest is called the transport time. Now, so we did experiments simulating this condition in our lab using a soil arena made out of plexiglass, and we had a soil as base and sprayed pesticide all over the arena at recommended dose. Now we have a colony called as the old nest. We moved the lid and put light stress, and they have a choice of a new nest where they can move. So, and we also have control sets where pesticide was not sprayed. Now, what would happen if the ants have to move through this contaminated environment? Will they be affected? Will they die? Will they be able to relocate? We found that 80% of the colonies in treatment groups successfully relocated to the new nest. But the time taken for them to do so was significantly increased, as evident by increase in discovery, latency, and transport time shown in orange bars. And why did this happen? Because most individuals were behaviorally impacted, as seen from this graph. That is approximately 60%, 50 to 60% of the colony were behaviorally impacted, and they could not do this process. Now, looking at the work distribution, only half the number of leaders were present who were doing this transport process in treatment colonies. So we can say that pesticide did impact their nest relocation, and situation might be worse in the natural habitat in presence of predators. In the next step, we asked what will happen on their nest choice. Say a colony is being kept in a contaminated environment, and they're given a choice to move to different sites with one contaminated, one not. So in order to do so, we used a T-maze setup in which we subjected control colonies, which was never seeing the pesticide before, to select from a nest which was either sprayed with pesticide or a nest which was not. Interestingly, we found that colonies which were not previously contaminated with pesticide selected non-contaminated nests, while colonies which were living in contaminated environment chose both nests equally. Now, tandem leaders, they are very important to do colony relocation, and since they are making so many multiple trips from the old nest to new nest, they are remembering the path so well. From previous experiments in our lab, we know that tandem leaders do have excellent memory, as we can see that 100% of tandem leaders could reach their old or new nest even after being isolated from the arena for 2 hours. Using the same setup, I asked if we would expose these tandem leaders with pesticide, will it impact their memory? Will they still be able to remember the target nest? And in order to do so, I wrote a grant, and I was lucky to receive a student research grant from Animal Behavior Society. What we found was in order to measure whether she was remembering the path, we had parameters of measuring success to reach either of the old nest or the new nest and path tortuosity. So we found in control, as expected, 100% of the leaders reached either of the target nest, but in treatment, 25% of the leaders failed to do so, and the proportion of success and in between control and treatment is significantly different. Next, among these treatment leaders who failed to reach either of the new nest had a highly tortuous path. That means they were not just staying there and not able to move. They moved across the arena, but they were unable to reach the target nest. Hence, we can say that the ants were being impacted by the pesticide, and it caused them to forget. In order to summarize, we can say that pesticide impacted the behavior, relocation dynamics of these ants, but the ant colonies were able to survive and recover from the effect. Working memory of tandem leaders was impacted due to the pesticide. I have submitted one manuscript and two are in preparation for me in this chapter. And finally, without the help of my supervisor and RPC members and my lab assistant and all my lab members, we are just like my ant colony. Without them, I would not be able to do this work. I would like to acknowledge my funding agency, CSIR, ISER Kolkata, Animal Behavior Society, and I would definitely like to acknowledge India Bioscience for giving me this platform to bring my science to you all. I'd love to hear your feedback and suggestions. Thank you.
Great talk. And Vidya and Tripura, over to you. Go ahead. Yeah.
I think I will just I will not be able to add too much of suggestions. It's totally off field for me. But I'm just curious, it's a nice and very, very interesting talk for me to look at that perspective. It's very different. I was just wondering, how do you pick up these tandemly? How do you identify them?
As in how do, so we actually visualize them. So for example, we have this big arena in our lab. So we can also, if you go outside and look at the ants, you can see that they start relocation. And if you look very closely, you can see the leader and the follower. And since these ants are 1 centimeter big in size, so it's not very difficult to look at them and take observations. Is that what you were asking, ma'am?
Yeah, I was just wondering. So there'll be only one, is it? In the entire group, there'll be only one tandem leader?
No, definitely not. Say a colony of 100 individuals are there. So one individual will start the tandem run and some other also will start. So approximately we know that 20% of the colony becomes tandem leaders. So there are simultaneous tandem runs going on across the colony.
Okay, so another very general question. How do you know the age of the
Sure.
colony? I mean, would these ants have, like all the ants in that particular colony have the same age, or each one is different age group?
We
Is
do
it the
not know, ma'am.
parents and they do not?
We collect the
Okay.
colony from nature, so we generally do not know how old they are. But if we want to do something about age, we have the pupa, and we can let some of the pupa to hatch into callows and then mark them and track them across days so we know their age.
Okay, but this kind of information, you pick them up from the wild, get them to the lab, and then you start looking at them, right?
Yes, yes. We do
And
not know actually how old they are when we are collecting directly from natural habitat.
And this pesticide spray that you use, so is it some impact of the concentration dependent? Because if it's a higher dose, will the ants die, or what is the dose? How do you decide on the dose that is to be sprayed on these colonies to understand?
Correct, right. So generally I wanted to study what happens in nature, and for that I wanted to simulate what people actually spray. So in order to do that, I read the label on the bottle and say they are saying put one whole bottle in 1 gallon and spray in 1 hectare. So I will standardize it for the area of my box and use the concentration and give the volume spray accordingly.
So you spray the entire, you were showing some images where you have the area. So you spray the entire place with pesticide. Is that
correct? So actually I can show you, I have these kind of atomizer bottles. So I fill it up and then I try to make it very homogeneous across the boxes.
And your
So,
control and your controls would be the ones which are non-treated. Similar age.
The controls are the ones I use water, tap water instead of the pesticide solution.
Okay, fine. Yeah, that's it. I'm not sure. I was more curious about these. How do you do it? That's very interesting.
I would love to answer more questions like this.
Yes.
Thank you, ma'am. Thank you.
Hi, Anvesha. Very nice talk.
Hi, ma'am. Thank you. Thank you.
I don't have very many questions. I missed something about what you said initially about no effects on colony sizes, I think. But did you also look at
yeah, sure.
reproductive output?
Yeah, I'll show you the slides again. I'm sorry, I might have gone too fast. So what I was saying is that we counted the colony size, which is before and after. So before being kept in the contaminated environment, how many were present, how many eggs, larvae, pupae were present. And since there is one reproductive individual called the gamergate, whether she was alive or dead at the end of 5 days. So there was no significant difference in terms of colony size or how many of them died, that is mortality rate.
So there was no effect on reproductive output also, or was there?
No effect on reproductive output in terms of eggs laid or survival of the gamergate.
I see. Okay. You also had something on the recovery of behavioral effects. Was that done on the same marked ants, or what do you think is happening?
This is a separate set. No, actually both. So this is a separate set of experiments in which I have 6 colonies lined together. All of them are definitely marked, and I take 3 hours of focal behavioral scans. So I will just take scans of how many ants are showing appendage shaking. I'll write, say, dash orange dash is said showing appendage shaking. Yeah.
Focals,
so I'll have it
in a scan. You might not get these events, right?
So these
you probably take focals, not scans.
Yes, focal sampling. Focal sampling of the 5 behaviors I have shown. So I will take that within the 3-hour window, and then I actually, for the sake of simplicity, I showed all of them pulled together in terms of induced behaviors, which is the graph below. And as you can see, on day 1 and day 2, the highest behavioral effects were seen, and slowly as the days increased, these behaviors just decreased. So say on day 5, I'm looking at a colony which was kept in 4 times the recommended dose.
Is that matched for individual?
It was, I have data for individual ants as well. I'm showing you for the colony. So I'll take the average of all the values of.
Whether you have data on individuals, because I was wondering how much is the turnover in the colony, and are you getting these effects because of that turnover, as opposed to the same individual's recovery?
So since I'm taking scans, I will not know if another individual is showing the behaviors in between when I'm not taking the sampling. So, but if I'm extrapolating it for say 3 hours, I might find the same individual doing the same thing for all across the 3 hours.
Okay. But you have the
is that
colony
what
that's not changing in composition over your study time?
Correct. Yes.
So
more or less all the individuals who are going outside and getting exposed are getting the behavioral effects eventually. So as you can see, average I'm showing, actually in terms of different behaviors, around 30 to 40% of the colony shows these behaviors at a point.
No, as long as you have the same individuals that you are looking at over time, it should be okay. Right? The colony is not changing in composition during your observation.
No, right? Definitely not. They are staying in the same box. They are not running away.
No, I didn't say in the same
and
box.
They are all
I'm
marked.
talking about those that you get the observations on. So in your focal,
right.
Are you sampling the same ants?
Yes. It depends on who are outside. So for example, I since they are living inside this nest, where is it?
No, I know the setup.
Yeah. So whoever is coming outside and showing the behavior, because inside the nest is very difficult to see who are doing what. Right? Inside the nest, if the roof is on, it's very difficult to see. So whoever is coming outside, I am taking the scans of those individuals.
Maybe you can take the common individuals across time and then see if they are actually recovering, or is this an artifact of something else? That might be
actually, ma'am, and on the 4th and 5th day, across all the scans, generally no one is being impacted. So,
correct. But the thing is, unless you account for the individual identity, how do you know that those individuals which are impacted are not remaining inside the nest and you're not seeing them?
Okay, so
maybe they're not
saying
recovered.
They might be showing these behaviors inside the nest.
Maybe they're very sluggish and they're not coming out. And the ones that are coming out don't show this.
Okay, so what from
So
my
if you take the
observations, I'm not
matched
showing the
observations
yes,
of the same individuals, then you'll
yeah.
be able to say for sure
Mhm.
that that's not happening.
Definitely. Yeah. We can do that analysis actually. Thank you, ma'am. I would try to incorporate it here. It will add a newer perspective, maybe like tracking the behavior across
Of
individual lines.
particular. I was also wondering, when
Yes.
you find those that live in contaminated environments choose both contaminated and non-contaminated equally, what's happening there? I mean, why do you think that's happening?
Ma'am, can you please
You
clarify
found
what you're
that
asking? I'm sorry, I couldn't follow
you
you.
found that those ants that were living in contaminated environments didn't show a choice for contaminated versus uncontaminated.
Oh, yeah, the
Right?
nest choice,
Right.
right?
Huh.
Yes.
So why do you
Yes.
think that's
Yeah.
happening?
My hypothesis is that they have been living in 5 days in this contaminated environment. They had the toxic effects. They showed all the behaviors, and after 5 days, they have almost recovered. Now, when they're again encountering, I think what is happening is behaviorally they don't. First of all, the first objective I would like to say is I don't think they can perceive that this is a contaminated environment. This is not. And hence they cannot make a clear choice. A. B.
No. But
The
they
ant.
should be able to perceive the difference between the two, because the ones that lived in the uncontaminated ones were differentiating. No?
Correct. Correct. Because they had never been in contact with this chemical. Maybe
Yeah.
the ones who are getting in contact of this chemical are getting, adapted to the environmental stress, and they generally, once they have moved to either of the nests, they do not want to shift. So if you look at this control colonies, initially they were split, like 3 of them were split and they could not make choices. And finally, after 24 hours, so this is a final graph, after 24 hours, the colonies who were split generally came to one nest site. So this was not a trend which we saw in case of treatment colonies. Treatment colonies just moved away from their nest and they just shift into one nest and they were like, we will stay here, we will not move. But we did not find anything like that with the control.
Very nice. I just have a final thing. In your acknowledgements, who was the Gamma
Yes.
Gate? You said these are all my people.
Well done, well done. Nice question, Anvesha. You want to hazard a guess? Yeah.
Oh, well.
You want to think about it and maybe use it in your next talk?
Yeah, I think so.
Brilliant question, Vidya. Great talk, Anvesha. Thanks
I
for the
have one
questions,
question.
Thank you.
Karishma, I
Vidya.
have one.
Yes,
Anvesha?
please.
Yeah, so my question again, you've been monitoring, like now I understand that you've been monitoring what, not the same individual, but you were monitoring them specifically whatever comes into their view, right? So any specific
Correct.
difference that you think, or have you been able to assess between the Gamergate and the other workers, or the impact of the pesticide on any of the behavioral studies that you've done?
So there are two components I would like to address here. First of all, in our species, we generally tend to think that the queen or the reproductive individual might be treated differently than others, but this is not the case in our model system. The Gamergate, even in tandem running, the relocation, the Gamergate is not given any specific preference that whether she will be taken
Right,
in
the queen
between or in first, nothing like that. And in my studies, I have not generally found any particular behavioral effects, any special, and I have not found anything with the Gamergate as a whole.
You have monitored the Gamergate separately. Have you done that?
Not separately. Generally, the Gamergate do not come out of the nest. She stays inside the nest. So she will directly probably not get in contact with the pesticide which is outside the nest.
Okay.
She will get indirectly contaminated by other individuals through physical contact.
So you would not
And
be able
in
to
one or two cases where I was in one or two cases where I could observe her inside the nest, I saw her showing the 5 different pesticide-induced behaviors, not paralysis though.
Okay.
I could see her showing appendage shaking, staggering, trembling in case of the box setup. But in case of relocation, where all the colony members were impacted, the Gamergate was showing all the behaviors equally.
Okay, then that's fine. All right, thank you.
Very nice. Good discussion, robust discussion, Anvesha. And Anvesha, a sign of a good talk is the number of questions. And you see the chat window. There have been so many questions. So I encourage you to now address them in the chat window if you have time. Very nice work.
Surely, surely.
Very nice work. Thank you.
Definitely, I'll do that.
Yes. You can stop slide
Thank
share
you so much, ma'am, for giving me this opportunity.
welcome, welcome. You gave us a bit of a heart skipped a beat. We didn't see you in the meeting room, but all's well that ends well. Okay. All right. So all the best. Our next speaker is Dimple Acharya, who's going to make us think about the malarial parasite and epigenetic regulation. So welcome, Dimple.
Hello,
Yeah,
ma'am.
yeah.
Am I
Welcome.
audible?
And yes, you're audible, you're visible. Why don't you put your slides on? You share your slides, Dimple.
Yes, ma'am.
And you have 20 minutes, Dimple. And
Yeah,
after
sure,
that,
ma'am.
10 minutes of Q&A. All the best, Dimple. Go for it. Yeah.
Thank you, ma'am. I hope my slides are visible, ma'am.
Yes, yes.
Yes. Should I go ahead, ma'am? Hello.
Yes.
Hello, Dimple.
Yes,
Please go ahead.
start.
Yes. Good afternoon, everyone. Thank you, India Bioscience, giving me opportunity to present my work on this platform. Topic of my PhD thesis is epigenetic gene regulation in the malaria parasite by DNA-based modification under the supervision of Dr. Shruti Vember from the IBAB Bangalore. So just giving you a brief introduction about my work. So my organism which I am working on is Plasmodium falciparum, which cause malaria. So it has a complex life cycle. It has a mosquito vector which will inject the sporozoite, and this sporozoite will complete the liver stage development and release the merozoite. These merozoites will invade the RBC, and in this RBC, it will complete its asexual development. The first stage of this asexual development is ring stage, which is a pre-replicative stage, followed by the trophozoite stage, which is a replicative stage, followed by the nuclear division through the schizophreny, and 16 to 32 daughter nuclei release into the bloodstream, which will invade the RBC and cycle continues. This is a 48-hour life cycle. And from this, some of the parasites will commit to form a sexual development and form a male and female gametocytes, which are ingested by the mosquito, and then cycle continues. Our lab mainly focus on the asexual blood stages because this responsible for the pathogenesis of the disease. And our lab mainly work on the how gene regulation plays an important role in the parasite's biology. So coming to the some of the key genomic feature of the parasite is it has a 23 megabase per haploid genome organized into the 14 linear chromosomes, highly edited genome. It has nearly 5,300 genes, from which 80% of its gene are expressed during the asexual blood stages, and it has a important virulence property, which is antigenic variations, which is regulated by the mutually exclusive expression of multi-gene families like VAR, Riffin, and Stewart's. There are two models known to explain the gene regulation in parasites. One is just-in-time transcription, meaning whenever protein is required, gene undergo the transcription and then translation, which is regulated at epigenetic and transcriptional level by the factors listed here, like DNA modification, nucleosome occupancy, etc. Another model is just-in-time translation, meaning mRNA has been already made, but whenever protein is required, this mRNA undergo the translation, which will regulate at the post-transcriptional level by the factors listed here, like mRNA stability, specialized RNA binding proteins, etc. So focus of my study is what is the role of DNA-based modification in the epigenetic gene regulation during asexual blood stages. So before going into that, just wanted to share what are the common well-studied epigenetic DNA modification marks in other eukaryotes. So we all know that 5-MC is generally well-studied epigenetic mark in higher eukaryotes. 6-MA is also well studied in lower eukaryotes, but in higher eukaryotes, it is poorly studied. Apart from this well-known epigenetic DNA modification, there is another modification, which is a guanine base modification. So during the oxidative stress, what will happen? This guanine is a most susceptible base out of four because it has a lower oxidation potentials, which will form a 8-oxo-guanine, which if it won't repair, it will cause GC to TA transversion, and if it will accumulate in the genome, this will cause disease like condition cancer. So there is an enzyme called DNA glycosylase OGG1, which will remove this modified base through the base excision repair pathway. So this canonical function is well studied, but in past decade, people have studied that this OGG1 dependent 8-oxo-G pathway is act as an epigenetic gene regulation. So in this model, if we see during the oxidative stress, this 8-oxo-G recognized by the OGG1 DNA glycosylase, which will read this mark, and then it will recruit the transcription machinery at the promoter sites, and it will express the gene which will help parasites, so which will help the organism to protect against the oxidative stress and help in cells to maintain the homeostasis, redox homeostasis. So this is well studied in the higher eukaryotes. So we wanted to study the same mechanism in the parasites, whether it is present or not. Before going into the my study, just wanted to show you about the whatever general DNA based modification known in parasites. So here Miss 5-MC, which is well studied in other eukaryotes, has found very low levels. Rather than this, a 5-hydroxymethyl cytosine like mark is present, but its function is still unknown. One study reported the 6-MA modification mark in parasites, but its function is still not clearly known, and other DNA based modification has not been explored. So we are going to explore the guanosine based modification. So based on this study, we have hypothesized that so during the oxidative stress, since parasites is growing under the RBC, which has a highly oxidizing environment, if 8-oxo-G will form, which could be read by the OGG1 and which can do the two potential roles. One, it will remove the 8-oxo-G through the base excision repair pathway, or it will recruit the transcription machinery, and it could be regulate the some of the genes of the downstream genes, which is related to the oxidative stress, and help the parasites to prevent against the oxidative stress or not. So to address this research gap, we framed the two broad objectives. First one is understanding the genome-wide distribution of 8-oxo-G in Plasmodium blood stages and its correlation to gene expression. And second objective is elucidating the role of 8-oxo-G repair enzyme OGG1 in Plasmodium gene regulation. So coming to the first objective, since we don't know that 8-oxo-G is present in parasites or not, so by using a southwestern blotting, and using the anti-8-oxo-G antibodies, what we have done is we have harvested the parasites and extracted the genomic DNA from different stages, spotted on positively charged nylon membrane, and developed against the anti-8-oxo-G blot. What we have observed here, when parasites grow from ring to trophozoite to schizone stages, this signal intensity has increased. And when we quantify by using the image J, we have observed that schizone stage has highest 8-oxo-G level compared to the other stages. This method confirms the presence of 8-oxo-G. However, further confirmation using a HPLC is in progress. Once we have confirmed the presence of 8-oxo-G, we wanted to know that where this 8-oxo-G is present in the genome wide. So to do the genome wide distribution, we use the method called oxidipsy. So this is the overview, general workflow for the oxidipsy. So we harvested the cells, extracted the genomic DNA, we break down into the small pieces, and then we denature. So that's how this antibody will work. It will recognize the 8-oxo-G mark when we denature it, and then immunoprecipitate it using the anti-8-oxo-G, and then eluted and purified, and next step is sending for the NGS sequencing. So since we are first time doing the oxidative sick in this organism. We were not sure that if this method is working and it is really enriching the 8-oxo-G containing fragments. So before sending for the sequencing, we did the quick dot blot method to check the whether whatever IP'd 8-oxo-G containing 8-oxo-G fragment. So here we have seen that IP'd 8-oxo-G eluate contains the highest signal intensity compared to its input and the no antibody control, which is suggesting that this method is working and it is really enriching the 8-oxo-G containing DNA fragments, not any other. After that, we prepare the DNA libraries and send for the NGS sequencing, and we analyze the data. So this data is showing. So this is the IGV screenshot of the enriched peak. This is the immunoprecipitated versus no antibody control fold enrichment peak. These are the input versus enriched and input versus no antibody control peak. So here we have observed that the peak which were mapping to the genome, it is present in the intergenic region of the genes in many chromosomes, which is suggesting that 8-oxo-G is present within the intergenic region of the genome. It's further correlation with the other data and gene expression like chromosome accessibility, nucleosome occupancy, RNA-seq to check that whether this 8-oxo-G peak are correlating with the gene expression or not. It is in progress, and generating other replicates and during the other stages also is in progress. So once we will get to know about this, we will know that which are the proteins, which are the genes are targeted by this. So this is the future perspective of this objective. Once we have confirmed the presence of 8-oxo-G, we wanted to know that is there any enzyme or is there any protein is present to read this mark and associated with this 8-oxo-G modification. So we have found the human DNA glycosylase homolog in the parasites, which is encoded by this gene called PFOGG1. It is a putative DNA glycosylase. Compared to the human OGG1, PFOGG1 is larger and its active domain is split into two. It has a big 200 amino acid insertion, and when we overlay the human OGG1 structure on the PFOGG1 active domains, it is overlaying nicely with the human OGG1. Apart from this, when we did the phylogenetic analysis, it is showing that this DNA glycosylase are specific to the Plasmodium species and they are forming a separate clade from compared to the other organisms. Once we have identified the homolog, we wanted to characterize this protein in vitro as well as in vivo. So to do the in vitro analysis, we have cloned the full length, means different version of the recombinant protein. So first one is full length, then artificially link both the domain 1 and domain 2, individually domain into the PET28A vector to tag with the his and to do the affinity purification. So despite multiple attempts, we couldn't able to purify the full length since it is high molecular weight, and always it should be in insoluble form and all. This kind of difficulties we have faced. So, but we wanted to study that this protein has activity or not. So we were reading literatures and all, and then we came across to this paper. In this paper, they have shown that methyl transferase activity by using a bacterial genomic DNA as a readout. So we adapted this strategy from this paper, and we used the bacterial genomic DNA to show that DNA glycosylase activity of PFOGG1. So this is the quick workflow how we did this assay. So we have individually transformed all the plasmids with all the construct into the BL21DE3 cells, and then we did the IPTG induction and expressed the proteins. After expressing protein for 1 hour at this concentration, we did the protein expression analysis by using anti-His or anti-PFOGG1D2 antibodies, and genomic DNA, means 8-oxo-G level of genomic DNA we use by anti-8-oxo-G antibodies. So here just wanted to mention that we could able to purify the individual domain, and we have generated antibody against this domain too. So after doing this assay, what we have observed here, so this is the PET28A control. When there is no PFOGG1 expression, we have seen that 8-oxo-G level has increased upon the IPTG induction compared to the uninduced genomic DNA. And when we have expressed the protein, that is D1 linker D2 or full length, we have seen that compared to the PET28A, decreased level of 8-oxo-G here. And same we have observed with the individual domains also when we express the protein. We have quantified this 8-oxo-G blot by the image J, and we have observed here that compared to the PET28A, we have seen the decreased level of 8-oxo-G when we express the domain 1 linker domain 2 domain individual domains. And GFP as a negative control. So this assay is conforms the DNA glycosylase activity of PF-OGG1. So once we have confirmed the activity, DNA glycosylase activity of OGG1, it has lyase activity also. We wanted to study its functional role inside the parasites, means in vivo. So to do the in vivo characterization, we have generated some strains. So first one, this is the P. falciparum wild type strain called 3D7 strain. Second one is the we have generated this strain. It is expressing GFP tag PF-OGG1 episomally in the parasites through its endogenous promoter to do the protein overexpression. And we have generated another 2 strain, which is in which we have deleted the domain 1. We have deleted individual domains, domain 1 or domain 2, by using a CRISPR-Cas9 system to disturb the PF-OGG1 domain. And then we did the phenotypic characterization. We did the protein localization, blood stage growth, oxidative stress response, check is 8-oxo-G level, and what is its effect on the transcriptome of the parasites. So here I'm not going to show the protein localization study, but we did the immunofluorescence assay of this strain, and we have seen that this protein, Miss PF-OGG1 GFP, largely localized to the nucleus. Then coming to its growth assay. So what we have observed here, when we overexpress the PF-OGG1 compared to its control strain, which is empty vector growing under the same drug pressure, it is growing better compared to its control strain, which is suggesting that having a extra PF-OGG1 provide a fitness benefit to the parasites. In case of the mutant strains, we have observed they are growing more or less similar to the wild type strain. So actually from the plasmodB data, we know that this protein is not essential for the blood stage growth, and we are observing the same trend here. After this, we have check the we have treated the parasites with the 4 different oxidative agent and see what is its effect on the growth. So this is the data for oxidative stress assay. So here we have treated with this 4. One is hydrogen peroxide, terbutyl hydrogen peroxide, potassium bromide, and menadione. So these 2 are the oxidative agent, oxidizing agent. KBRO3 will increase the 8-oxo-G level in the mitochondria, and menadione is just generate the superoxide, and it will affect the electron transport chain of the cell. So here what we observe that overexpressing strain of the PF-OGG1 is growing better when we expose to the oxidizing agent of terbutyl hydroxide and menadione compared to its control strain. In case of mutant strain, we haven't seen much effect. They are growing more or less similar to the control strain, which is suggesting that maybe parasite. So in the absence or in the disruption of the PF-OGG1 function, parasites will use some other DNA glycosylase to overcome this effect. After that, we have checked the 8-oxo-G level of the strain. So here, our expectation is when we overexpress the PF-OGG1, we should see the decreased level of 8-oxo-G. So here we have observed the decreased level of 8-oxo-G compared to the control strain in case of ring and trophozoite stage, but not in trizone stage. But in the mutant strain, our expectation was when we disturb the PF-OGG1 or decrease the PF-OGG1 level, we should see the increased level of 8-oxo-G. But here, in sometimes it is behaving like increase the level, but sometimes not. So this result is inconclusive. We are also thinking that what is happening here. So we are currently process of generating some more replicates and all about this. So this result is inconclusive. apart from this, we have studied the effect of what is its effect on the transcriptome. So in when we disturb the domain 1 or domain 2, so this is the workflow. We harvested the parasites at two stages, ring and trophozoite, because we wanted to study its effect on transcriptome. So in these two stages we harvested, then prepared the harvested RNA, and then prepared the library, sent for NGS sequencing, and then we did the quality analysis. So this quality preliminary analysis shows that this ring stages samples are making a different cluster from the trophozoite stage, and all the ring stage samples, mutant as well as wild type,
Sorry
are
to
clustering
disturb you,
together.
Dimple.
Sorry.
Sorry to interrupt you, Dimple. Your 20 minutes of the talk are up. You can wrap up.
Yeah, this is my last slide. Yeah. And these trophozoite samples are making a separate cluster, and they are mutant are forming a separate from the wild type. So we did the DE-seq2 analysis, and we observed that during the trophozoite stages, we have seen the more differentially expressed gene in both the strain, and it has almost 50% overlapping differentially expressed gene. And then these are the some of the important genes, like oxidative stress related genes, thyroredoxin, respiratory gene, and cytochrome BC1 complex. These are upregulated, and some of the genes which are related to the gametocytogenesis processes are downregulated. So this data is suggesting that when we disturb the OGG1, it is affecting the other stages. So might be it is not important for the asexual stages, it will important for the later stages. So just wrapping up, my conclusion are, we have detected the 8-oxo gene, and genome wide data are suggesting that it is present within the intergenic region. The in vitro study suggests that PFOGG1 has a DNA glycosylase activity. Overexpressing the PFOGG1 decrease the 8-oxo G level, and disturbing the PFOGG1 affects the parasite transcriptome during the trophozoite stages. Significance of this study is, this is the first time we are reporting the guanosine modification, and will this emerge as a new epigenetic layer, and we will get to know that which genes these are targeting, and what is its role in telomere biology. At last, I would acknowledge my supervisor, my lab mates, and my funding agency, DBT JRF, and India Bioscience once again for giving me opportunity to present my work. I would like to have feedback from the mentors.
Thank you for your talk, Dimple. It was very well presented. I now hand over to the expert mentors.
Yeah.
It-
Yeah, Vidya, please go
I
on.
don't have any comments. I think Tripura will handle that. It looked like a lot of work.
Thank you, ma'am.
Yeah. Hi, Dimple. So
Hello, ma'am.
for me also, this is a little different area, but then I have some questions which I would like to first understand. So this seems actually a very interesting study, because we are still trying to understand, TETS itself is new, trying to understand the 5MC, 5HMC in different things itself is still, we are in the infancy stage. This is definitely a very new finding, and I'm quite surprised I'm hearing it for the first time, and I think it's a very good work. That first of all, I should congratulate you. It's a very good piece of work.
Thank
But I
you, ma'am.
have this, some questions. I want to look at your the cloning experiment where you've done different
Ma'am, in vitro study?
yes, in vitro. Yes, exactly. So
Ah,
here
yes,
you said you were not able to get the for the full length, right?
yes, ma'am.
And you've removed the but you were able to get the rest of the 3.
So, ma'am, for the domain 1 linker, domain 2 was also insoluble, and we were not able to purify it. Only individual domain. So, domain 2 we have successfully purified first.
So I wasn't able to quite link up with this, the PFO GG1 act. Correct me if I got it right. So this PFO GG1, this gene is, the protein is responsible for removing the oxidation state on the OXOG, is it?
So, ma'am,
Or
it will remove the modified 8-oxo
modification. So
as well
basically
as it has lyase activity also, ma'am. So it will create a nick also.
it will create a nick, and then it will remove this base modification. So what is the next base which as a readout? So is that what are you going to use as a readout? If say, for instance, if for 5-HMCs there we have 5 fluoryl carboxyl, we'll have a next stage.
Yeah.
So here, is it only that, 8-oxo-G is removed and then there's a nick and then there's just a replacement of normal guanine? Is that how it is?
Yes, ma'am. We are looking for this kind of assay from this one.
Okay, so what is the link between the bacterial genomic oxygen? Is that used as a readout? Why did you go for the bacterial one? Why not use the same DNA of the malarial parasite? Maybe I'm missing some point there. Could you please clarify that?
So, ma'am, actually, we for the doing the in vitro assay, if we are using a genomic DNA of parasites, we want active protein for this, but we won't able to purify this. So what we did is we just transformed the plasmid only and express the this one in the bacterial system
So this is
and
in
check.
the E. coli system. You're expressing, and in this itself, you're trying to understand as a
Yes.
functional.
Yes, it
what
is. Yeah, we should see some defect in the 8-oxo-G level with some effect on the 8-oxo-G level. So here this is empty plasmid, ma'am. It doesn't have any protein like UGG1 and something. We have seen that IPTG concentration, means IPTG induction only causing a stress and it will increase the 8-oxo-G level. But in case of other, when active domain like D1 linker D2 is expressing, we can see the decreased level of 8-oxo-G compared to the PET28A. When protein is expressed, we have seen, like, we have confirmed protein is expressing and then we are doing this.
So this is done in the E. coli system because it's easier
Yes.
to understand this one. So you're still developing this as a readout system, as an in vitro readout model, or is it already established?
So, ma'am, these people have already they have used first this system to to show that methyl transferase activity.
Okay, so now you want to understand which domain is responsible for this function among that was the main question you want to understand.
So here we miss individual domain also, plus major thing is, does this protein has glycosylase activity, which does it affecting the 8-oxo-G level? So first was this, and of course we wanted to understand the individual domain function also.
You did not want to try any transient repression by shRNA or something, siRNA something in the host itself. You just do transient repression.
So actually, ma'am, our parasite doesn't have RNA interference pathway.
Oh, okay.
Yeah, that is our limitation.
Fine. All right. Okay, get it. All right. So I think it's good, very good work and quite interesting and very recent. I think it will still. I think you're going the right track. I'm sorry, I won't be able to add more because I'm more on curious note than understanding what exactly it is. So I will wish you lots of good luck, and I'm sure this will be a very, very interesting piece of finding.
Thank you very much, ma'am.
Thank you, expert mentors. Definitely, it was a lot of work and it was very well presented in terms of figures and graphs. Thank you, Dimple. Now you can stop sharing. Okay. Our next speaker is Anirban. Anirban, can you confirm with your audio and
Yes, I'm here.
great. So Anirban will be talking about the crucial role of miRNAs in bone formation. You can start sharing your slides, Anirban.
Yeah.
Okay, you have 20 minutes for your talk.
Yes.
Okay.
Yeah, I hope my screen is visible right now.
Yes, it is in sharing mode.
Sure. Hello everyone. Good afternoon. I am Anirban Sardar, working right now in CSR CDRI under the supervision of Dr. Ritu Trivedi. Here I would like to present my recent work entitled Lactational Osteo-miRNA plays a crucial role in bone formation. Coming to the literature review, in the normal condition, normal physiology, there are the serum calcium level that are coming influx into the bone and bone act as a reservoir, and that it comes out to the serum level. There the homeostasis is there, and along with the kidney, the entire homeostasis of calcium functions in the normal physiological condition. Whereas the disturbance in the calcium homeostasis observed during the cases of pregnancy and the lactation. Especially in the lactation, the serum calcium outflow from bone is getting higher as there is the requirements of a baby. and there is a very higher amount of calcium depletion from the kidneys also there. That leads to the decrease in the overall calcium level in the bone and making some cause in bone disorders and increase in the serum calcium level. At the molecular level, basically it is the prolactin hormone that is functioning, and it's known to replace the gonadotropic releasing hormone in the hypothalamic gonadotropic releasing hormone, and thereby decreases the gonadotropins like LH and FSH, and thereby it reduces estrogen and progesterone, the female hormones, and thereby along with the prolactin, it targets bone cells and decrease the calcium level and increase the serum calcium concentration, and thereby hamper the bone formation. There is another major player, that is PTHRP is also functioning during the lactation phase. This PTHRP is also known to cause calcium release from the bone, and thereby it replaces the function of calcitonin. And in the clinical sample, it was observed that there is a depletion in the total calcium level, calcitriol level, PTH, prolactin, estradiol, PTHRP, all are getting regulated during the all phases of reproductive cycle like lactation, weaning, and the post-weaning. Here I have listed some change parameters like serum calcium, albumin free corrected calcium, ionized calcium, all are getting regulated in the cases of human as well as in the rat and mouse. Herein, I am interested in the lactation phase, and due to this, the trabecular bone loss of about 50 to 10% in the case of human, whereas 25 to 35% in the rat and mouse is observed, and due to which it is a alarming concern, and somehow it is neglected. And therefore we are interested in interested to check the miRNA profiling of during this phase. And for this before the screening, we try to identify what is the actual phase where the bone loss are predominantly observed, and the screening of the screening data observed that at the age of day 18 of lactation, the increased amount of bone loss, the highest amount of bone loss was observed, which was found recovered at after the day of day 60 in the mice sample, and this was also observed in the cortical bone as well as in the trabecular bone. And this data was then we have screened out the microRNA. We have micro profiled the microRNA. What are the to understand the what are the differentially regulated microRNA during this day 18 and day 16. And we have identified several upregulated microRNA which are shown to upregulated during the weaning, and some downregulated microRNA which was observed during the weaning. Out of all these, I am interested to observe the effect of MIR 204. Since we have already established the role of some upregulated microRNA that is known to play a crucial role in the bone formation. Herein, I am showing the data to elucidate the role of MIR 204, which is found downregulated during the case of weaning. And to validate this data, I have checked the expression of free MIR 204 in the bone sample of nulliparous animal and lactation. The increased amount of free MIR 204 as well as mature MIR 204 was observed. It was around 10-fold increase mRNA expression was observed in the bone samples of lactating animals as compared to its nulliparous counterpart. To further confirm this, we have observed that this MIR205P was shown in the bone cells that like BMSCs, that is bone marrow stromal cells, osteoblasts, bone forming osteoblast cells, as well as cartilage forming chondrocyte cells. In all three cells, this positive expression was observed. That indicates this microRNA is present all over the bone. And when we try to understand whether what is the status of this MIR2045P in the bone forming osteoblast cell differentiation. Osteoblast cells usually differentiated in the case of bone formation, and throughout the differentiation, we observe a sharp decline in the expression profile of the MIR2045P. To check what the exact role of MIR204 is doing in regulating bone formation, we have introduced a commercially available MIR204-5P inhibitor to inhibit the exogenous miRNA, and MIR204-5P mimic to overexpress the miRNA. And the data indicates that after treating osteoblast cells with MIR2045P inhibitor, MIR2045P expression was getting downregulated, and after treating the cells with MIR2045P mimic, the expression of MIR2045P was upregulated. With this strategy, I tried to to evaluate the osteoblast differentiation by checking alkaline phosphatase activity after treating cells with inhibitor and mimic and compared with their respective negative control. And the data observed after various time points indicate a sharp decline in the osteoblast differentiation after treating cells with a mimic, while the significant upregulation in osteoblast differentiation was observed when cells were treated with the inhibitor. This indicates that miRNA MIR2045P may show have potential role in bone formation. This data was further validated through staining with the ALP, and after this exogenous in vitro bone formation was tested for these cells after treating cells with inhibitor or mimic. Cells were stained for Sirius red staining to observe the calcium deposition and alizarin red staining for the mineralized nodule formation. As a hallmark of bone formation, both of these staining data showed increased profiling when in the cells treated with the inhibitor, while a repressive bone formation was observed when cells were treated with the mimic in both the cases in Sirius red and the alizarin red staining. The quantification data also indicate the same. After confirming the role of MIR204 in osteoblast differentiation, we try to evaluate the molecular regulation of this miRNA in mediating osteoblast differentiation. For this, we have check the expression profile of certain osteogenic markers like RANX2, ALPL, BMP2, OCN, and COL1A1. All these tested gene was found elevated when cells were treated with the inhibitor, and it was found replaced when cells were treated with the MIR204-5P mimic. The test results was also confirmed in mRNA level via qRT-PCR, as well as in the immunoblot experiments for the protein expression data. After confirming the molecular regulation of MIR204 via certain markers, we are now trying to narrow down what are the exact targets of this miRNA. As we all know that miRNA specifically targets some mRNA to exert its function. For this, we have employed bioinformatics and observed and screen out from the commonly available databases like TargetScan, MadeDB, or Picture to identify the exact target. And from all across all these three, we have identified a RANX2 and LP as a common target from these three well-known databases and tried to evaluate whether this miRNA is targeting RANX2 in LP in the biological system or not. For this, luciferase assay was executed. And for this, we have cloned the RANX2 wild type sequence as well as RANX2 mutant sequence in the luciferase vector. And similarly, for the ALPL gene, the wild type as well as the mutant sequence was cloned in the luciferase vector. And then the cells were co-treated with MIR2045P mimic to check what is the luciferase activity. The test results indicate a significant decrease in the luciferase activity when cells were treated with the RANX2 wild type plasmid along with mimic, while no such change in the luciferase activity was observed when cells were treated with a mutant variant of any kind of identified gene along with MIR2045 mimic. That indicates identified sequence as a main seed sequence for the target binding of the identified miRNA. After confirming this, we have employed a shRNA to partially knock down the target genes like RANX2 and ALPL. For this, we have used the commercially available shRNA that is already available, and the data we also try to evaluate whether this shRNA is actually performing its potential in our system, in our test system. And it was found a significant decrease in the expression profile of RANX2 when treated with the RANX2 vector. However, for the ALPL, we need to screen out because there was no available shRNA in the market. And for this, we have used two commercially available shRNA and test for their efficacy. From this screening, we have used shRNA, one of the shRNA for the test system in our next experiments. And from after the screening of shRNA, we have checked the osteoblast differentiation after co-treating with the MIR2045 mimic by assessing the ALP activity. ALP here again indicating the osteoblast differentiation status. And the test results indicate when both the shRNA was employed, the repressive osteoblast differentiation were noted. However, after mimic treatment, this was further reduced, and which indicates that MIR-2045P targets the identified mRNA like RANX2 and ALP to exert its function. Then to further confirm these, we have tried to evaluate the expression profile of tested gene like LANX2 and ALP in the similar system at both mRNA as well as in the protein level. The test results indicate a repressive action when ACE was employed as compared to negative control. However, after mimic treatment, it was further reduced, which indicates again that MIR-2045P targets RANX2 and ALPL to impede osteoblast differentiation. This data was further validated in the in vivo system. For the in vivo system, we have initially observed, we have executed primary osteoporosis model. For these, animals were overectomized, and we have checked the expression of MIR-2045P and the MIR-204. Both the expression profile was found increased in the overectomized animal, in the osteoporotic animal as compared to this healthy control. However, both the target was found repressed in the osteoporotic animal as compared to this treated animal. Similarly, for the secondary for osteoporosis model, we have injected animals with dexamethasone, and then bone samples were examined to check the expression profile of MIR-2045P as well as the pre-MIR-204. Both the expression was found upregulated in the osteoporotic sample as compared to the healthy control. However, like primary osteoporosis samples, both the expression of the targets was found repressed in the bone samples of osteoporotic animals as compared to this healthy control. After confirming all these as an outcome of osteoporosis, skeletal fracture is there, and we try to employ the role of this miRNA in the skeletal fracture. For these two different animals were bone were fractured, and then expression of the miRNA and its target was checked after 5 days of fracture as well as the 10 days of fracture. At both the time point, it was observed that MIR-2045P was found upregulated, while both the target was found repressed like in the osteoporosis, and this lead to the confirmation that MIR-2045P may play a major role in bone loss condition and skeletal fracture. To confirm this, we have employed some statistical model to check the correlation of MIR-2045P with its target like RANX2 and the ALP. The data obtained from the primary osteoporosis, secondary osteoporosis, and fracture indicates a positive correlation in the expression of MIR-2045P with its target, and this indicates that there may be a possible role of MIR-2045P in regulating bone formation via this identified target like RANX2 and ALPL. In summary, from the lactating mother, we have identified MIR-204, which is known to target RANX2 and LPL, and by targeting these two, it actually target cellular differentiation of osteoblast cells, and thereby contributing to the pathogenesis of osteoporosis. And here I would like to acknowledge my supervisor, Dr. Ritu Trivedi, my lab members, Animal House, CDRI, and obviously the heart gratitude to the animals. Its contribution have been integral part of this study, and India Bioscience for giving me the opportunity to share my results. Thank you.
Thank you, Anirban, for the wonderful talk. Now we can move to expert mentors for suggestions and feedback.
Yes.
Yeah, hi, Anirban.
Hello, ma'am.
So, I have one question. You've done this you did some cell culture studies in MSCs.
Yes.
Bone.
I did yes, yes, ma'am, bone cells in MSCs as well as in osteoblast and osteoclast and osteocyte as well as chondrocytes.
Okay, so these MSCs were from the mice itself,
Bone.
right?
Yes, yes, ma'am.
And you did with the NIH3T3 as well. It's a known osteoblastin, right? Do you check that, or you did you did only in mice?
Only in mice. Yeah, no, only in mice as well as in the cell culture, but we isolated cells from animals, not the cell
That's
line.
what I'm asking. So your cell culture also was only in animal model.
Yes.
So the initial data that you had shown, the initial introduction, that does this the same miRNA play a role in human cells as well? It would be maybe interesting to just check in any
Yes,
other.
ma'am. Yes, ma'am. The data I have not shared, but yes, for the human studies, I have employed FOP 1.19 cells to check the role of this miRNA in human osteoblast. And yes, the similar kind of data I have observed in human cells as well. And one thing is very interesting to note that the target that I have identified, the RANX2 and LP, the seed sequence is basically all are same across the kingdoms.
So
I
it's
mean, from
all
human,
conserved. So the same,
yes, all are
these are
conserved.
the same targets which are seen even in humans you are
Yes, yes,
checking.
ma'am, yes, ma'am,
Is
yes,
this
ma'am.
MIR 2045B, has this been implicated in any other conditions or any other tissues? Or
Yes,
is it
ma'am. Yes, there are some reports that indicates it known to cause cancers and like prostate cancer and all.
it's involved in cancers?
Yes,
The
cancer diagnosis.
lack of this causes, or is it overexpression of this causes
It
cancers?
overexpression cause the cancer.
Um, so would it not be interesting? Probably. Did you do any overexpression studies with this? I saw that
For the overexpression, I used miRNA mimic and to check the role of
that
this
was the
miRNA.
mRNA mimic that you
Yes,
used.
yes, ma'am.
And that was a transient expression, right?
This is transient expression.
So could you try anything with the instead of transient? And transient would be there only for you did the lipofectamine based transfection?
Yes, ma'am. Lifetime.
That
Yes.
was very transient for 2 or 3 days, probably, right?
2 or 3 days, but I have checked this expression throughout for 21 days because one of our experimental protocols suggests that I need to culture cells for 21 days to check the in vitro bone formation.
Induction. Yes, yes.
Yes, I need to induct multiple times in that case.
Between you were doing the induction of this, right? In between the experiment.
Yes, ma'am.
So you were, but this one, if I understand, the 21-day protocol is a continuous one, right? You don't
Yes,
just.
ma'am.
So how were you?
Yes, ma'am. But after 3 days, like you said, after 3 days, I have to induct cells with the miRNA mimic or inhibitor, and thereby I continued the culture for 21 days. I mean, 7 treatment for 21 days.
Okay. So how do you, because when we do this lipofectomy, it's pretty sensitive. We need the cells to be in a particular stage when they take up the RNA, right? So
Yes,
during
ma'am.
the induction process, they're completely differentiating, right? They're maturing basically. So how sure are you that this overexpression has, I mean, was there a continuous overexpression or both knockdown overexpression for 21 days, right? So
21 days, yes.
how did you confirm that, at those points your lipofectamine based transfection was successful? How do you confirm? How do we know?
Yes, ma'am. For this, I have checked through TrackMan assay, and the TrackMan assay suggests that depletion in the expression when I used inhibitor and the increased expression when it was used for mimic.
Okay, so you've done it at 0
Yes,
point
ma'am.
and in between, or just 21 days again?
In between as well as at the final time point.
Harvest in between and you check that.
Yes,
okay. Sure enough. I think I don't have any specific suggestions. You're towards the end of your PhD?
Yes, ma'am. Actually, I have defended my
I know,
thesis in on
it's
20th
more or
only.
less complete. That's why I was just wondering that you were presenting here, because you've addressed all the points, and that's very interesting. So you did this overexpression studies even in the human cells, or you just checked for the levels of this microRNA?
I did the overexpression study I have done.
Okay.
And from the database I collect, there are some databases available in the PubMed, and I have also confirmed the expression of these in the osteoporotic sample of humans as well from the other authors definitely.
Okay, that's a good piece of work, Anirban. But I only felt just a suggestion that, objective should have been spelled more clearly, because you directly went into the data. I was looking more for the objectives to understand why exactly.
Yes.
I was trying to look for the lactation specific, those kind of mice data, and then suddenly you jumped into the data. So it wasn't little clear there, but of course we could follow later. But in your next talks, whenever you give maybe a little bit of introduction on the objectives, and because you start with lactation model and that is to present the case, but then you actually studies was on the that your actual idea was to look at the expression of this or relevance of this in bone, right? So maybe that just a presentation, just improve that on presentation of what is your clear objective, it would make it even more better. Otherwise, it's a good study. All the best, Anirban.
Thank you, ma'am. Thank you so much.
Yeah, I agree with Tripura, maybe a little more on the objectives, but it looked like a lot of work, and I don't have any specific comments.
Thank you so much.
So in one thing though, in your graphs, when you have these bars and those points, so are they 6 experiments or are they
All are
6
the replicas,
individuals?
ma'am. Yes,
Or?
ma'am. All are the individuals. All are the different individuals.
So they are the number of individuals.
Yes, ma'am. Yes, ma'am.
Okay, so each individual is counted as 1 point, and it's one
1 point.
experiment with 6
Yes,
individuals.
yes, ma'am. Yes, ma'am.
Okay, so do you also repeat the experiments, or I mean, are these 6 individuals
Actually, ma'am,
used
the
in the experiment at the
for,
same
you
time,
asked for
or
the
how is it?
animals. You asked for the animals or the in vitro data. In the case of in vitro data, we experiment different at the different time points. For animal study, definitely it is in one go, but different animals.
And then what statistics do you do on them?
Ma'am, all of the I have used ANOVA followed by two case.
Anirban, by any chance you look at the serum levels in human samples? Any chance that you could get to look at this microRNA levels in the serum of patients with osteoporosis?
Ma'am,
Or any
that
information already available?
no, I was looking for the databases is absolutely correct, but to get human samples, you also know to get a human sample,
I
it is
know it's not for your PhD, but
it's
as a
bit
continuation, it would be very
yes, ma'am.
interesting to look at
Yes, ma'am.
patients with the serum samples. You can look at for
Yes, ma'am.
osteoporosis patients specifically. I'm sure you'll find a lot of them in the clinics.
Yes, ma'am.
It's just
Absolutely.
serum based, so I don't think ethics should be much of a problem. That would be
No,
very
ma'am.
interesting to check on this.
Yes, ma'am.
Even to compare in the lactation as well as in the osteoporotic patients, that will be a good correlation to understand.
Yes, ma'am.
All the best.
Very nice. All the best.
Thank you, ma'am.
Thank you, Anirban. It was an interesting talk to know the role of lactational miRNAs in bone formation. Thank you so much. Now
Thank
we'll
you.
move to the long-awaited disclosure of our giveaway. We have a winner, and Prashant Bhagwati is our winner. He has won the book. Prashant, are you in the meeting room? Okay. So if you are in the meeting room, please email us at hello@India Bioscience. Now, towards the end of the session, I thank all the expert mentors for their priceless suggestion and feedbacks. And thank you to all the PhD speakers for taking us through the vast diversity of animal biology. Thank you so much. And this was our animal biology session. So tomorrow we are heading towards our 4th day of PhD Cafe. We will be having 2 sessions for tomorrow. It's membrane biology and medicine and physiology, sorry. So please be there. We will be waiting for your presence. Thank you so much.
Yeah, I would like to thank India Bioscience. It was normally we don't get to see such diverse topics in different areas. It was especially I was fascinated with the ant study because that is something which we never see in our forums. It's more of this what we see of gels and clonings and all that. So that was a very interesting thing. I think this is a very good initiative, and I would appreciate India Bioscience for this.
Definitely. For me also, I'm a nature enthusiast and plant insect interaction. So it was very interesting talk of ants for me too, how the behavioral biology. Yeah, yes.
Thank
Thank
you.
you so much. Bye.
Thank you.
We'll see you.
Thanks. Bye.